Right, thank you so much, Dr. Jander, for that introduction. And so, as you said, I'll be talking with you today about my work in the Harrison lab looking at arbuscular mycorrhizal symbiosis. So to start off, and let's get the Okay, so to start off, as many of you probably know, phosphorus is a vital macronutrient for animals as well as plants, involved in things such as energy transfer through ATP, as well as membrane integrity through phospholipids. So when plants are trying to get it, the form that they often get it in is this inorganic phosphate form that's found in the soils. And this form is not very mobile in soils. It's not able to move around as readily in soils. It's not as soluble. So what you get is roots that are surrounded by a phosphorus depletion zone. They take up all the phosphorus around them, and that's not able to be readily replenished. And then you have this phosphorus sufficient zone that's outside of the area accessible by the roots. And so what this means is that when we're putting phosphorus fertilizers on our fields, we're not being able to get the full extent of their use out because phosphorus is not readily available to the roots. They use it up and there's a depletion zone that's created. And the problem with this is that the phosphorus fertilizers are non-renewable resources. We're using them a lot quicker than the minerals can be replenished. So a lot of work is looking into ways that we can maximize their use. And additionally, when you have excess phosphorus in the soil, many of you may have heard of phosphorus runoff that leads to eutrophication of waterways. And this is an image of a toxic algal bloom in Lake Erie, suspected to being caused by phosphorus runoff from agricultural fields. So a lot of work in this area has been looking at how we can maximize the efficiency of phosphorus intake of plant roots, and especially accessing this phosphorus sufficient zone to make sure that all these fertilizers we're putting on the field are being used to their fullest extent. So part of this work has looked into the discovery of arbuscular mycorrhizal fungi, which can extend the phosphate availability to plants. So what these fungi do is you can see these little yellow tendrils, that's the fungal hyphae. They can extend into the phosphorus sufficient zone and uptake this inorganic phosphate. And then they can form and actually grow into plant roots. And what they do is grow into the root tissue and form what are called arbuscules and the cortical cells. So these branching structures, which are the site of nutrient exchange. So this is where we see that phosphate that the fungi has acquired in this phosphorus sufficient zone is being taken in and transferred to the roots. And then you also see that the plant gives carbohydrates to the fungi. It's a symbiotic relationship. And then, so a lot of the work in this area that has been done up to this point with the mycorrhizal fungi has been understanding their structure and understanding different transporters implicated in the transfer of these nutrients between the two. And this is just an image where the fungi have been stained, showing the arbuscules and the root cells. So some of the work that my work was to understand how environmental factors impact this relationship, which is something that had not been investigated as much due to a lack of technologies to be able to do it. So some of the things that we wanted to look for were what evidence do we have of different environmental factors which may impact the ability for this nutrient transfer to occur, or the nature of this nutrient transfer. So previous members of the Harrison lab had looked into understanding how differences in the mycorrhizal fungi that a plant is associated with impact the shoot phosphorus levels, the shoot phosphate levels. And what they found that there was a difference with some of these species. So there was some sort of correlation here between fungal species and differences in the phosphorus uptake. And so through my research, I wanted to ask if this correlation is also seen in root phosphorus levels, if we also see this relationship at the root level in the arbuscule specifically, where we know the fungi is interacting with the plant roots. And specifically, the Rhizophagus irregularis, one of the fungi in this study, is one that we took and used in the work that I did. Additionally, we had another question of seeing how nitrogen impacts this relationship as well. This is a study done in maize roots, no fungal association, but what they did find is that when nitrogen fertilizers are added, you have a higher rate of the phosphate intake. So there was a relationship here seen preliminarily between the intake of these nutrients. And additionally, some previous workers in the Harrison lab had found that nitrogen does play a role in arbuscular development. So we do have evidence of this relationship. However, we don't know the nature of it. And so we wanted to be able to measure if cytosolic phosphate levels differ in arbuscule, in the arbuscule symbiosis, depending on the nitrogen fertilizer treatments. So the crux of our work, as I had mentioned before, generally when you would go to assess phosphate levels in roots, you would have to take a whole root sample and grind it up and then measure the total phosphate in the root. You weren't able to do cellular localization. With a new technique that's been developed in the lab is very novel to being able to help us do this work, is we have a fret-based sensor that is expressed in the root cells. And this allows us to quantify locally the concentration, the relative concentration of cytosolic phosphate. So how the sensor works is you have a phosphate binding protein attached to two fluorophores. And when no phosphate is present, the two fluorophores are in proximity to each other. And so you get the excitation when you go to measure, you excite one of the fluorophores, then through fluorescent resonance energy transfer, you get a transfer of energy between the two, and you get emission by the fluorophore that was not excited, but had energy transferred to it. And so when phosphorus is present, you get phosphorus that binds to this binding protein, and then you see a conformational change that moves the two fluorophores away from each other. So when you excite the one fluorophore, you get no emission. And so through this, we're able to do a quantification where we can see that the phosphate level, as it increases with the sensor present, the fret ratio, which is a corrected measurement of the emission of this fluorophore, drops. And to really confirm and make sure that the phosphate level is what is causing this change, we also have a control sensor that cannot bind phosphorus. So we can see how the fret ratio may change non-specifically due to things like pH or other environmental conditions in the cell, so that we can really make sure with our phosphate sensor that the relationship we are seeing is due to changes in phosphorus concentration. So to begin this investigation, we had our little cones here, and they were filled with a sand mixture for root growth and easy root extraction. And then we had a fungal spore layer, which was added. And in this case, we were looking at the fungal species, the differences that causes in the cytosolic phosphate levels. So for this, as I mentioned before, we looked at Rhizophagus irregularis, and then we had Diversospora epigea, which is another species of AM fungi that has been studied in the lab. And then finally, we used transgenic Brachypodium distachyon, expressing either the phosphate sensor or one of our technical control sensors. And this plant's used because we know that it has these fungal associations, as well as it has thinner roots, which are a lot easier to image with confocal microscopy, which is what we used later. So we were able to get clear images with that. And then so the plants were grown for 3 to 4 weeks. And then we had fluorescent imaging with a confocal microscope. And then this is an example of what the images would look like for plants that had arbuscules, so had those fungal associations. This area, you've got the epidermal cells out here, which are not colonized with the fungi. And then you have in the middle here cells where you can see their arbuscular development. You can actually see some of the hyphae growing through here. So those are the regions we would look at for the phosphate sensor, phosphate sensor levels. And then, so what we discovered with this is that there was a difference in the phosphate sensor between the two fungal conditions. And there was not a difference in the control sensor between these two conditions, which is very important. So the fact that the control sensor was the same between the conditions allows us to localize this difference we see in the phosphate sensor and be able to say that this was due to a difference in the relative cytosolic phosphate level. And so that is what we were able to confirm, that there was a difference in relative cytosolic phosphate level between these two fungal species, as the shoot data had shown before from another member of the Harrison lab. And then we wanted to go and look at the nitrogen's impact in a similar manner. So in this case, we did a with AM symbiosis condition with Diversospora epigea, and then we had a without fungal condition, again, just using this technique in general to be able to see cytosolic phosphate levels is very novel. So being able to confirm data that had been previously collected in other ways was another goal of this experiment. So these were grown for 4 weeks, like the other condition, treated with a low nitrogen fertilizer, and then we imaged the low nitrogen condition, treated with a high nitrogen fertilizer, waited half an hour, and analyzed took images of the high nitrogen condition. And unfortunately, we did not get fungal colonization with our AM symbiosis condition, so we do not have data for that, but we do have data for the non-colonized without AM symbiosis condition. And then this is an example of what those looks like without the fungi present. So the just regular cells with the phosphate sensor. And so again, what we found for this was that there was no significant difference seen between the control sensors in the two low nitrogen and then the high nitrogen treatment condition, but there was a significant difference, and there was a difference in the low nitrogen and the high nitrogen treatment with the phosphate sensor. And in fact, going along with the data that the research group had found previously with the corn roots, they found that there was a drop in the fret ratio, which, as I mentioned before, correlates to an increase in the relative cytosolic phosphate level. And so we were able to confirm then this relationship again, using this novel technique, showing that a high nitrogen treatment leads to an increase in the relative cytosolic phosphate level. And so overall, the study, the work I was able to do this summer did confirm that there are important environmental factors that impact the rate of this nutrient exchange between mycorrhizal fungi and the plant roots. And then with the nitrogen treatment, that there was a relationship between these two factors. So there was a difference in the relative cytosolic phosphate levels between the different fungal species conditions. And then there also was no data that we got for the AM symbiosis condition, but we do have data confirming previous work showing that nitrogen application increases the amount of relative cytosolic phosphate levels. So future work, again, taking this novel method that we now have to localize in the cells and in the cells specifically that contain arbuscules. A lot of future work is going to be just data collection and understanding these relationships further and gaining out. So for example, with the fungi species, looking at more of the fungi species that we have to understand what the differences may be between them and the rate of this nutrient exchange. And then also looking to complete our data set to go back and hopefully get fungal growth so that we can see how nitrogen impacts cytosolic phosphate levels in root cells that have AM symbiosis. And then finally, a little bit more of a downstream application as we are looking to use this in the fields. One of the important things may be to understand the mechanisms through which these occur so that we can better utilize them and maybe tend them towards helping our crops to more efficiently get the phosphate that we are putting on them. And so with that, I would just like to thank the NSF and the USDA, as well as Cornell University for allowing me to be able to be here today. And then a really, really big thank you to everyone at BTI, specifically the members of the Harrison Lab. So Dr. Harrison, and then my mentor Shiki, as well as all the other members of the lab for welcoming me into your space and allowing me to do some really, really cool work this summer. So thank you. Yeah.
Possible commercial aspects of this research, especially like applying it on an industry-wide scale? Do you think that this could become eventually commonplace?
Well, it actually, so there are companies that do sell mycorrhizal fungi currently. One of the big things with that, though, and especially looking at an agricultural scale, so utilizing this in fields, I think, is the eventual goal that I see for it, especially as I had mentioned with being able to, fungal hyphae are able to extend a lot farther than plant roots can. Plants are a little hesitant to go so far just because of how much it takes from them to be able to grow super expansive roots. But so part of this, part of the problem is a lot of change in the agricultural practices would have to happen. So like tilling the soil obviously would break up fungal hyphae that have been in place. And so there are companies that do sell fungi. And so this work in understanding environmental impacts can kind of help us to see what practices we may have to change in order to use this on a much wider scale.
No further questions. Thank you.